So kind of to motivate this discussion, one thing that is super important is actually looking at your data. Four, to the eye, visually different examples, but that all have the same summary statistics. And so, a lot of times you learn a lot more just from eyeballing and looking at your data than maybe what a model can even tell you. But with that being said, data visualization can be hard. Rendering needs to be fast, and a lot of times you have a lot of high dimensional data that you're trying to visualize, data from different streams. You might be looking at behavior, you might be looking at some electrophysiology or calcium imaging or whatever methodology that you're using to collect your data. But all those things, they don't have the same sampling rate. They need to be visualized efficiently and easily, and that can be really challenging. Also, a lot of current visualization libraries in Python, it is really hard to make interactive visualizations. Lots of plots that you get are static, or you have to learn like new infrastructure and API in order to make interactive plots, which can also be really, really challenging. If you have ever tried to simply view your behavior, and for example, here we're showing calcium imaging data, just scroll through in time and see your movies in correspondence with whatever neural data you recorded, it can be very, very challenging. And so kind of also along that vein is also, why don't scientific plots look as good as modern video games? On the left, you're kind of seeing this game from about 20 years ago compared to, a game today. And clearly there's been a huge jump in the graphic technology from 20 years ago to now. And why haven't we really seen that change in scientific visualization? What kind of things are they doing that really just hasn't been brought to our arena? And so that's kind of what fastplotlib aims to do. It uses new graphics technologies, the same as what modern video games are doing, in order to be able to create really high-level interactive visualizations. And so these are just some examples of some cool stuff that we've done before. We've done some point tracking with some models from Google. We've done a lot of neuroscience analysis, and then also just some fun examples to kind of show you and introduce the library as well. And we'll get into some of these later. But, I kind of just want to spend the next couple of minutes talking about some of maybe the more technical details. So the next couple of slides are not all that important for using the library, but just to kind of give you an idea of where fastplotlib is at. We're going to propose that we have a task. And in our task, this is going to be very simple. We're just trying to plot this one 3D RGB image. And, obviously, for all of you in this room, your task, whatever data that you're working with, is obviously a lot more complex. But just to keep it simple, at the top of this, kind of ecosystem of what fastplotlib is built on top of is us, and it would take about 4 lines of code to do this. You define a figure, you have your data, you add your data to the figure as an image, and then you show the figure. And so the reason that we really even built fastplotlib is to kind of fulfill this need of we're scientists, we want to focus on our data. And, at these lower layers of abstraction that I'm going to go through, it takes so much more lines of code, and you get to focus less on your data and more on the code, and as scientists, that's really not what we want. And so we're built on top of PyGraphics, which is a rendering engine. And so, as you kind of go down the stack, PyGraphics and WGPU are kind of bundled together. These developers out of the Netherlands have done some really amazing work to bring those to fruition. But, at the PyGraphics level, to plot this image, it would take a lot more lines of code. And you have to define things like the renderer, the canvas, the scene. And maybe this doesn't seem like that many lines of code, but this is just for an image. You could imagine as your visualizations become more and more complex, that this would become even more difficult and honestly tedious and feasible, and not really what the goal of what you're trying to accomplish is. And even lower beyond that, to do this in WGPU or more like the hardware level of code, it would take several hundred lines of code, which is, ridiculous. And so really what allows fastplotlib to work and what, kind of what this stack is built on and, is really leveraging these new graphics APIs, and that's Vulkan, Metal, and DirectX 12. And really what they do is they leverage the modern GPU hardware better than OpenGL, which is kind of like the old standard, and which is what a lot of older Python visualization libraries that you're more familiar with are built on, like VisPy, Matplotlib, they all use OpenGL. And so we're kind of making this transition to these newer graphics hardware, which really allow us to have these powerful interactive scientific visualizations. And so remainder, we're going to spend our time at the fastplotlib level, which is where we really care about, and it's roughly 4 lines of code. And so, what is fastplotlib? It's a high-level API for scientific plotting, inspiration from PyQtGraph and some other libraries. We're built on top of the PyGraphics rendering engine. This project is very new. We've only been working on it probably for like 2.5 years. And the goal is fast visualization via an expressive and elegant API. We want you to have your data in the structure that you're used to, in an array-like interface that all you're doing is interacting with arrays, that you don't have to learn some crazy new API or data structure in order to make your visualizations. We want you to think less about how, to define your interactivity in your visualization and more about, what's the best way that I can view my data. So, what can I do with fastplotlib is GPU accelerated visualization. And honestly, for most use cases, the integrated GPU that you have in your laptop is sufficient. So what you're seeing in the bottom right is a visualization that Kushal and I did very early on when fastplotlib was just coming about on his old laptop GPU. And what you're seeing is 3 million lines, or 3 million points, 10,000 lines, I think. And it's very performant. And then in the top right is actually streaming data that someone's reading off of a biological device. And so it's great for rapid prototyping and algorithm design, which Kushal has been doing a lot of work with for his PhD. Time series exploration if you want to work with machine learning models. The goal also is exploration and fast rendering of large-scale data. A lot of times your data can be terabytes in size, and how can you visualize that really quickly and easily? And also, real-time acquisition systems for instruments also could be useful. And this is not an exhaustive list, but this is, some of the cool stuff that we've been able to do with fastplotlib that it's just not easy in other libraries. Also, where can I use fastplotlib? The nice thing about it is that you can write your code in whatever arena environment that you want and run it anywhere. So it's nice if you want to prototype in Jupyter Lab and then, go on to run it as, an independent web application or in Qt. It's the same code no matter what, which is also really, really helpful. And so now I'm going to turn things over to Kushal, and he's going to walk through some actual demos or some examples of how we've used fastplotlib.
Okay, so Caitlin just gave a great technical overview of the new technologies that make this possible. And I know that, a lot of you are perhaps, experimentalists. I used to be an experimentalist as well, so I know what it's like to, work in a wet lab and what you really want out of a software tool. So I'm going to try and guide you through some examples of, what does all of this mean? What does this let you do? So let's just start simple. So this is something that Phil made, within a day. I showed him how to plot lines and do a few simple things in fastplotlib one day, and then I came back the next day and he had this full visualization up and running. What Phil wants to do is he's tracking worms and analyzing their behavior in an arena, and he wants to verify that the tracking that has been done makes sense. And that's something that you do a lot in behavioral experiments, or anytime you have an instrument and you're doing any kind of recording, you want to verify that that your measurements match your raw data, and that's what Phil does over here. So what we specifically have over here is, so in the top right you see the video of a worm crawling around in an arena, and then over here on the left is the worm track, and the color indicates the ethogram. So a different color means it's doing a different behavior at that time point, is it moving forward, is it doing a turn, is it moving backward, things like that. And the white ball that you see moving around is the location of the worm at this time point. And then on the bottom right over here is an odor concentration with the same ethogram mapped onto it. And Caitlin is actually going to demo, how exactly you can create this visualization. But we've tried really hard to make it easy and fast to implement things like this. So next, let's go to something a bit more complicated, where you're performing some neurophysiological recording, like in this case calcium imaging, and you are simultaneously recording behavior. Anyone who's tried to visualize or analyze or synchronize data like this knows that it can be, it's non-trivial, and it's even harder to be able to look at all of this data in sync. And sometimes it's hard to even just look at the data, in terms of visualizing it like this. So what you're seeing here is, it's a video of a live render. This is not something that's like rendered and then saved as a movie. You can actually interact with this. You can zoom in, you can drag things, you can click on things. So it's not like it's not like a slow library where you render things and each frame takes like a second to render. This is all done on the fly. And what you're specifically seeing here is, for those of you who may not be familiar with calcium imaging, so these left two panels on the bottom are basically two different views of a field of view from calcium imaging. These contours is encircling a neuron, and what you can do is you can explore through this data. You can click on different neurons, and then the temporal activity of that neuron is then highlighted up over here. What you see down here is the ethogram, which is the behavior that the animal is performing at that particular time point. And then over here you have an overview of all of the traces. This vertical white line tells you what time point you're currently at, so that that corresponds to what the current image frame over here looks like. And then down here you can also see exactly what the animal was doing at that time point. And finally, on the right over here is a PCA of the calcium activity, where the white ball indicates the current time point. So what this allows you to do is, for example, you could be like, oh, I see something weird here in my calcium imaging trace. Let me see, let me click there and see exactly what the animal is doing at that time point. So, it really lets you, it's a way that powerful visualizations like this can actually, drive scientific discovery. It's not just like, oh, let me, make a plot. This is something that you can actually use and iterate with and prototype to, drive your scientific questions forward. So a more powerful example of just that is something which I do. So what we have here on the left is what's called a covariance matrix of a calcium imaging video. And the goal of this is, okay, so I work with someone who's an amazing applied mathematician. We work together really well because he'll come up with ideas, and he'll be like, oh, can we look at this covariance matrix? And he'll, oh, can you print the first row? Can you print the second row? I'm like, we can just visualize all of this. But it's things that you typically don't think about, because there are a lot of things which are just usually not possible. You can't you can't plot something that's 3,000 by 3,000 and interact with it easily. Okay, 3,000 by 3,000 is actually pretty small. You can plot things that are like 100,000 by 100,000 pixels and still explore them. So what I'm showing over here is every single element in this matrix, so kind of like every single pixel in this 3,000 by 3,000 matrix is the similarity between like one pixel in a calcium imaging video with every other pixel. So this can be hard to interpret by just looking at the numbers, or by just looking at this matrix in the form of like a colorized heat map. What's more powerful is to look at each of these rows and actually reshape them back into an image. So what we have over here, for example, is, if I pause this, so there's this row, and that row corresponds to this pixel that's brightly lit up over here, and the brightness in this image represents how similar is that pixel's activity to every other pixel in this frame. And this is a much more powerful way to try and understand whether some algorithm that you're working on or some method is giving you things that make sense or not. Okay, and lastly, this is something that we found that other people are using fastplotlib for. So I found that Facebook Research or Meta is actually using fastplotlib for live streaming of VR data. So what they have here is they have cameras in a VR device that has a live video stream, and they're synchronizing that with some other time series sensors that they're also recording from, and they're able to stream all of this data and visualize it immediately. Okay, and a last example is fastplotlib will work as as both like a desktop application, but you can also use it to create web apps with the exact same code, and that's what this demonstrates. And powerful is, like, let's say for example you're about to publish a paper and you have like 100 gigabyte data set, or like 100 gigabyte video, or in this case like a 3D reconstruction that you want to show to people. It can be hard to transmit that data, and then the person on the other end trying to recreate the visualization. So like, why not just give them the visualization? And that's what fastplotlib allows you to do. So what I'm about to show you right here is a live example of a I think this is like a 150 gigabyte data set. And the thing is, we don't have to transmit all of that data to us in order to look at it. So this data sits somewhere on a server, and we're just streaming the visualization live. And this is something that I can interact with fast. Okay, if I know how to use your mouse. How do I click and drag? Okay, well, we can go through. Okay, yeah, I do not know how to use your mouse. Sorry, can you?
Maybe it's with the server?
Is the browser?
I don't know. I'm on the guest Wi-Fi here.
Okay, that's okay. Okay, well, it is playing. Your mouse drag is not working. Keyboard events might work. Okay, that's okay. How do I click to get to the other tab?
What are you mean? Bottom left.
Okay, I think your touchpad is off. Okay. Okay, we might show you that later if I can. Okay. Okay, I'll give it back to Caitlin.
Okay. Okay, so next I'm going to spend some time going through some demos, actually showing you how to use fastplotlib, but just kind of to give you an intuition of how the library is kind of designed. So you have a figure, and that can be two options. So option A on the left is that you only have one subplot in your figure, and you can add graphics to that figure. But then we also showed you, some examples where there was, more than one cell of things going on. And so that would be option B on the right. You have multiple subplots, and then you can add graphics individually to those subplots. And then the interactivity you kind of define between the graphics, between subplots. But that's kind of just intuition, off the cuff, an idea of how the library is laid out. And is this a good size? Should I increase? Can people see the font? Okay, good size. Okay. So we're going to be using some image.io examples, and these are also like on a public repo on fastplotlib, like that you can download and play around with yourself. We also have, I'll go through at the end some documentation that we have and some examples. But essentially, remember from the very beginning when I had the simple task, we're trying to plot an image. Well, we're going to replicate here with a different image, but basically we're defining a figure. And by default, our figure is just going to have one subplot. If we don't specify how many we want, you just get one. And then we're going to have our image data from the image.io library. And then we're going to basically take our figure, add an image, give it our data. We can also give our graphic a name, not that relevant here, but we're naming it sample image, and and then we show. And so we're actually using, maybe my mouse isn't working.
There's a pop-up at the bottom.
Maybe I should try. I have my actual mouse. Oh, I think maybe because I'm stepping on the cords. I think that happened earlier. Okay, let's see if it works. Oh, here we go. Okay, I'll just use my mouse. Maybe my trackpad is messed up. I've had problems with this laptop. Okay, so we get our figure, and we're using Sidecar, which is this like nice Jupyter Lab like library utility that like you don't have to use, but we're going to be doing some updates, and so it's nice because when you scroll down and run your code, your example stays on the side. But basically, we get our image. We can move our image around. We can zoom in, we can zoom out. We have these nice, like, auto-scale the scene. You can center the scene. You can, click off so then you can't move it and it stays fixed. We can also turn off maintaining the aspect, which means we can, like, stretch and compress our image. Or, we can leave it how it is. But so then graphics have what we like to call, these properties or graphic features. And that means that, like, after you plot something, it's mutable. We can make changes. So, for example, we can dynamically change the color map afterwards. We can also kind of look at like what the underlying data is. And so this is just like a 2D 512 by 512 like pixels. We can also get a view of that data. So here we're looking at, the X coordinates like pixels 10 to 20 and then every fifth one of the Y. And so that's kind of, what you see here as an array form. And then we can also do fancy indexing. So we're going to take every 15th X and every 15th Y and change that pixel value to 1. And so it kind of creates this like hatching pattern. We can also do something even more complex. We can say, like of our image data, every pixel that has a value greater than 100, set it to 255. So that's going to like whiten out our background. And then we can also add events. And so that's what allows you to define kind of this interactive plotting that we showed you kind of in the examples. And so here what we're going to do is add an event so that, when we click, we're going to see a red dot appear. And so that's kind of just, simple example, intro, how do I define a graphic? How do I add it to a plot? How can I have some interactivity or interact with my graphic after I've plotted something? And so next is, I want to show you guys the image widget. And so if you work with time series data, this can be really, really useful because you basically just give it your image data and then it automatically generates these sliders for you that allow you to scroll through your data. And so first we're going to be looking at this movie from the image.io library, which is like this cockatoo. And so basically we automatically get this time slider for us that, updates and scrolls through time, very easy, nice. But, say I had like multiple dimensions. And so here we're going to be looking at this whole brain zebrafish data, and we've kind of taken like just a subsample of this data. So it's normally, I think it's normally like 30 gigabytes, like 32 planes, and then you get like through time also, but here we're just doing 100 frames in 4 planes. And so we can kind of create 2 options. So on the left, what you see is we get 2 sliders. And so we can scroll through time as we were before, but right now we're only looking at plane 0, or like the first plane, but then we can also, change what plane we're looking at and also scroll through time. But then we could have passed in our data to the image widget differently and gotten each plane as its own unique subplot, and then scrolled through time and seen all 4 planes together. And that was really like the exact same code basically. The difference is just in how we passed in our data. Here we passed in everything, so it made 2 separate sliders, and in the second figure on the right, we passed in, each subplot as its own in a list, and so we only had 1 slider for time. But basically, the image widget, kind of handles creating all of those things for you, so that you don't have to have the repetitive UI code of you having to over and over write, oh, I have a slider, and every time that I move the slider, I want it to update the frame that I'm seeing. And so next, I want to show you some cool stuff with line plots. And so to start, we're just going to generate some data. And so we're really just using a sine and a cosine wave. And then we're going to create a figure and add those lines to the figure. And so in the top, we see our sine and cosine wave. And we gave one wave. We just said we want the color to be magenta. But then you can also do something cool. You can imply an entire color map to your line. So in the top, I think we chose autumn. But we can do our fancy indexing just like we did with our images. And so here, with our cosine graphic, for the first 30 points, we're going to select every other point and change the color to cyan. And so that's kind of what you see in the top here. And then we can also add an event handler to say, okay, if our colors and our cosine graphic change, let's print the event information. And so now, if we take our cosine graphic and we change, the last 10 points to blue, what we get printed out is to say, here was, the slice or the key that was used, and we changed the user value I passed was blue, and here's what the new value is. And so just like we assigned the color map to the top line, originally it was autumn, and now obviously I've done, some indexing to change some of the colors, but I can also very easily change the colors of my sine graphic, and also apply a color map to it, so now it's seismic. And what's really nice is then, we have this color map, but say, sine wave, we've got values above 1, we've got values below 1. What if we wanted to color our line based on the y values of our sine graphic? We could very easily use what we call a color map transform, to which then you're assigning specific values. So now, this divergent color map, at 0 we see the white, and then our values that are positive. Above 0 we see red, and below we see blue. And so you can very easily do that, and you might have as part of your data some type of qualitative metric that you're trying to assign colors to. And as I'm going to show in Phil's data in a few minutes, behavioral ethograms is one key example of this. If you've got some type of classification, you've run it through, you've got colors you're trying to segment and with your video, having this flexibility in being able to easily assign a color to a certain data point can be really, really useful. And so last, we also have some selector tools. And so here what we're doing is we're just adding a linear region selector. So, and what you're seeing is the zoomed in data in the bottom. And so, I can stretch and I can kind of move it along and kind of edit what part of the data that I'm looking at. And so if anyone in the room has ever done EFIS stuff, a lot of times you're recording for so many hours or minutes or whatever, and maybe all of the information that you care about happens in a 1 millisecond time bin. And so being able to kind of zoom in and look at a specific region of interest can be really important. And lastly, I just want to show you guys some scatter plots. And so basically we're defining these 3 clouds, and they're just some basic scatters, and we can do the same things that we were doing before, where you can move things around, whatever. And so we can do all of the same fancy indexing and slicing that we were doing before. So we can change colors, we can change positions of the data, we can also define animation functions. And so an animation function is just a function that's going to get called on every single render cycle. So every time that your data is fetching to the GPU, this function is getting called. And so we could kind of make the clouds jitter in effect, because all that we're doing is like every render cycle to like when it goes to the GPU and like fetches the data buffer, it's saying, okay, update my data like this, and it gets called, and so like our points look like they're kind of just, you know, jittering back and forth. And you know, we can also do something like cycle the colors, and so like our one cloud, all that's happening is every render cycle, it's like cycling through like this ring of colors and changing. And so that's kind of like brief introduction. I'm sorry if that's fast. Sometimes I talk too fast. But that's kind of like all the different types of graphics and kind of things that you can do with graphics in fastplotlib, and like very introductory. But now I want to kind of move on to like more scientific examples, because I think that that's more relevant. But we have like a similar like intro type notebook like in our documentation that kind of walks through these things also, that like if you're, you know, trying to install the library and just get started with it, like that's a good place to start in terms of like kind of getting your mind around how the API works. And so now we're going to recreate the example that Kishal kind of went through of Phil's data. He was nice enough to send this to me last night. And basically, it's the same thing. We're going to look at the worm and then like the behavioral ethogram and kind of the concentration plot as well. And so to start, we can just kind of like view the worm. Oh no, it didn't save my work earlier. Sorry. Okay, sorry. So basically, this data set has like 3 different worm data in it. And so this is the first worm. And, you know, we can scroll through time. We're using the image widget just like before, so we just give it our data, and it automatically creates a slider for us, and we're in business. And so here's one worm, and we obviously, I think I chose just the first track, but we could have done anything. And so, you know, if we're looking at, say, the next track, you know, we define a track, and then, you know, we have our worm viewer, and then we can also, you know, do something where, you know, say we want to look at the corresponding behavior. So, you know, we create a figure, we haven't added anything to it, and then Phil gave me this color dictionary, which kind of defines in his mapping, like what behavior is going to go to what color. So there's like forward run, short turn, forward curving, omega pot, like in general, I don't know what these means in terms of worm, but this is maybe behavioral markings of the worm, and we're basically just saying, oh, we want, you know, forward run to be red and short turn to be blue. And, you know, we kind of go through the behavioral options and, you know, kind of like predefined those. And so then like within Phil's data, every, you know, like data point or every point in time has like this behavior is forward run. And so we can very easily, you know, take our color dictionary and say, you know, create a list that has, you know, at every point, you know, give me what color that's going to be. And so this is just the first 10. So the first 10 are gray, so unclassified to start. Our worm, we don't know what it's doing. But, you know, if I printed the whole array, there's like, you know, like 10,000 time points, so it would just go on and on. So maybe I should have chosen a more interesting segment. And so basically now we're going to take our XY position of our worm and add that to one of our figures. And maybe I should show, yeah, maybe I should show you what it looks like so far. Okay, so what we see so far is we have our worm track, and it's colored according to the behavior, and then we also have our worm video. And then what we define after that is our concentration plot, which is like the Ode curve that Kishal was showing from before. And we also just want to add an event handler to say, okay, as I slide through time, update my plot to what I'm looking at. And so now we see, hold on, maybe I zoom out now so you can see better. Okay, so concentration plot on the bottom. We also have our XY position of the worm, and then we have our image of the worm. And then as, you know, we scroll through, oh no, my mGUI went away. Oh, I had this issue earlier. Oh, I think it's when I have same plots open. Okay, sorry. I have my worm, and I scroll through time, and so we see our dot who's tracking our time position, and then it's also connected to this linear selector. So everything is synced up so that as I scroll through time, I'm seeing what my worm is doing. It's in the gray right now, so we're saying unclassified behavior, and then, we can also see our concentration plot. And, we kind of adjust and scroll through time. And, this is really good for, trying to see, is whatever behavior classification that I did, is it factual, like, does it match up? So if you're trying to assess, like, maybe whatever method that you use to get the classification, if that's what your interest is, or just in general, to kind of eyeball and see, okay, I have my worm, is my worm doing, what is it doing? And so that was Phil's data, and so I thought that was really cool because I've never worked with worm data before, so that was really nice. And then last but not least, we are going to go through another example, sorry, the demos, but essentially the next data set we got from someone at Flatiron, and it is they're looking at like head direction cells, and so what you're going to see is this mouse that's kind of like on this platform that has different arms, and like the mouse like goes on the different arms and like tracking its head angle as it's on this track. And what can we do to like kind of investigate the tuning curves and some general like neuroscience ephys analysis that is done. And this is also using Pineapple, which is this other open source Python library that like works with time series data, and so we're kind of using that in conjunction with fastplotlib. That's just like a side note. And also this time store that Flynn has also been working on in the interim, but that I kind of like worked on over the summer. But essentially, like, it helps you synchronize your data in time because we're gonna have our behavioral videos as well as our calcium, and like those occur at different sampling rates, and so it's really nice to be able to make sure that everything is happening synchronously and that our visualization is getting updated as we want it. And so we have our behavioral data and our calcium data, and like clearly those are not the same amount of time points. So clearly something needs to happen in order to make sure that when we say I want to look at time at 5 seconds, that we're getting 5 seconds in our behavioral data and in our calcium data. And so also one thing that happens a lot, especially with calcium data, is that you can get this like Vinet effect, and so in order to make like the quality of the video look better, you can kind of subtract out the minimum frame. And so all we did in what you're seeing here is like, what is the minimum frame within the calcium movie, and then when we plot our movie, we're gonna like remove that, so like basically subtract that from every frame, and it's going to like remove this Vinet effect. But just to kind of show you like what that looks like. And so super helpful if we just look at what the calcium and what the behavior looks at to start with. And so on the left you see our calcium, on the right you see our behavior, and I can kind of move through time, and as you see the mouse kind of is like going down these arms of the track, and there's like some kind of reward at the end of each arm, and then we have in conjunction our calcium, and those are nicely synchronized. And so one thing that Kushal showed you in the example from before, if you're not really familiar with calcium imaging, is like you want to identify the neurons and the components, and so we can kind of calculate those and then overlay them onto our calcium imaging video. And so clearly we've identified some components. And as we're kind of like looking through this, like maybe these components that are like on the top right or on this like kind of like outer edge, like I don't know, maybe some of them are not very good, like so like the next thing you do is like maybe filter out some of the components so that they look, maybe you're only getting like whatever metric you use, like the good components. And so, based on the head angle, which is like kind of the task or like kind of what we're interested in investigating here, we can kind of sort the neurons like good versus bad based on like which ones we think correspond most to head angle. And then, I can kind of identify. So a lot of the ones that are white, which I'm considering like my good neurons, and the ones that are bad are like my pink neurons in terms of how we sorted them. And like, the ones that I visually saw that were, maybe I thought were maybe not all that good of neurons, I'm going to remove those.
How did those blobs, were those user-defined ROIs, or was it a cell segmentation?
Yes, they were already segmented in this data set, and so I just made them into contours and plotted them. Like, you would have done, you would have used like Sweet2P or Cayman or like some like segmentation algorithm in order to like get those ROIs to begin with. But here they already had them stored like in the data set when they were given to us for us to just plot. And so basically I can filter my data set and like only plot the good ones. And so now, moving forward, maybe we've identified some components that maybe aren't that good, and we can keep going forward. And so another nice thing to look at is the, look at the temporal traces, which is, kind of what Kashal had showed before. And so as opposed to looking at the traces as like individual curves, we're looking at them as a heat map. And so, like maybe, we can go down through here, and this would be like selecting an individual row, and then we can also, we can also move across time again, and so, we're synchronizing our selector, and so now, our behavior is synchronized with our calcium, that's synchronized with looking at our traces, so, we can identify a point in time that we really like, maybe there's lots of activity, kind of see what the cells are doing. And, maybe also we would want to like select individually like one of these components and like, see what it's doing. And also look at the tuning curve. And so now, I've selected this neuron, but maybe I select this one. Sorry. My clicking's not working. Oh, sorry. Maybe it's not clicking. Maybe I use this. Sorry. I select my component. I forgot which way I defined the interactivity. But I can select a component going like this, and then it's going to zoom in to that neuron, and then I can look through time and see, okay, maybe like when is this neuron active? And, what is the behavior? Where is the mouse? What is the head angle? Let's look at the tuning curve. What is the head angle at that time? And so, maybe I could do that to also, kind of investigate single ROIs. What are they doing versus like what's the head angle? And so next I want to show you just like in conjunction like the PCA space for this. It's kind of noisy, not all that informative, but just kind of to show you, maybe how you would look at that. We could also add a figure for the PCA. And so, here's our PCA space, and, we can kind of move around in it. But then, also, as we scroll in time, you can kind of see this white dot that also shows you, like where you're at in PCA space. And, it's kind of nice, you can change the controller type, and so then it kind of, just for fun, it looks like you can like fly into your data, which we think will be really cool for like astronomy examples, not necessarily maybe all that cool for this one, but just to show you that that's like a possibility. And so, that's kind of like, obviously I didn't go line by line to show you like what all the code like individually is doing, but this is kind of like how you could maybe build up a like complex visualization for neural data that shows you, how I would maybe go about investigating maybe a question of interest that I have. Like I have these head direction cells, how can I really see, if what, my hypothesis is and confirm. And so, that's how fastplotlib can be used to like kind of drive scientific visualization and like drive your scientific questions forward. And so in terms of demos, that's all that I have. And at this time, if there are any questions for Kashal and I, we would love to answer questions.
What's the dependency landscape? How big of how many dependents? Is this
Yeah, so we actually just did a release this week. I would say that in terms of like stability, like we are built on PyGraphics, which is like this Python rendering engine, and they are not quite yet stable. And so like we will become more stable as they become stable. But I would say in terms of like the API and like the things that you can do with fastplotlib, like those are all concrete. Like the features that we're supporting right now, like we're going to continue to support. In terms of like the size of dependencies, I feel like we try to keep it to a minimal. And if you kind of like go through our installation guide, you can kind of see what options. Like if you don't want to have like mGUI, you don't have to. Or if you don't want to use fastplotlib in a notebook, like you don't have to install the dependencies for the notebook. If you would prefer to use like Qt or GFLW, like you have kind of like options in what kind of dependencies you want or not. I hope that answers your question.
No, and just to add on to that, it's like WGPU is very